Good morning, everyone. I'm Simran Da Costa. And on behalf of Altum Technologies, I'd like to welcome you all to this virtual event on bioactive lead molecules identified in Artemisia scoparia and their anti-Theileria equi-efficacy. So, before we begin, a little bit on Theileria equi. So, the disease equine pyroplasmosis is a tick-borne protozoan disease of equids and is characterized by pyrexia, intravascular hemolytic anemia followed by jaundice, pale mucous membranes, edema, tachycardia, occasionally thrombocytopenia, and finally multiple organ failure leading to a fatal outcome. And currently, the only drug available for the therapeutic management of equine pyroplasmosis is imidocarb dipropionate, which is relatively safe but may cause systemic toxicity like colic. So, what we are going to look at today is the entire layout of the study that was planned to evaluate the in vitro anti-Theileria equi activity of the Artemisia scoparia plant extract and also to identify the various the various phytochemicals and lead molecules present in it. So let's understand more about the study from our speaker of the day. It gives me immense pleasure to introduce Dr. Sanjay Kumar. So, Dr. Sanjay Kumar is a principal scientist at ICAR, National Research Center on Equines, Hisar, India. He has done his PhD in veterinary medicine from CCS, Haryana Agricultural University, Hisar. He has done his postdoc from National Research Center for Protozoan Diseases, Obihiro, Japan. He has an MBA from Punjab Technical University, Jalandhar. He has worked extensively on serodiagnostic tests for Babesia equi infection and on immunoprophylactic activity of Babesia equi merzocytes in donkeys and identification of immunodominant polypeptides. From the year 2010 to 2013, he was the principal investigator of OIE twinning NRCPD Japan, NRCE India on equine pyroplasmosis. He has a membership in several high profile academic boards, and he has also been a part of many national as well as international projects that mostly involve studies on equine pyroplasmosis. So, without taking much time, I'd like to hand over the platform to our expert, Dr. Sanjay. So, welcome once again, sir, and you can take over from here.
Thank you, Dr. Simran, for introducing me. Uh, Good morning to everybody. I'm just presenting here some of the work of my PhD student, and we were working on some plant, and we extracted the plant molecules and then tested it against the Thleria equi. So let me tell you about something about the. This is the protozoan parasite of animals, and most of us have heard the name of the plasmodium. It is malaria, and that malaria is a mosquito-borne disease. And similar type of protozoan parasite is in animals also, and we call it Thleria and Babesia. It is here, you can see it, and it is tick-borne. If we all know the what are the ticks, they are small creature like the small arthropod rather, like the mosquito, and it is small arthropod. They suck the blood from the animal, and then while sucking they transmit the parasite. And in this Eimeria, Coccidia, Toxoplasma are other epicomplexin parasite. So we can say that in it is equine malaria parasite. In layman language we can say it is equine malaria. So this is the intra-erythrocytic. It is how it is the inside the red blood cells. These are the Maltese cross-like, and these are the merozoites. These are the merozoites, and these cycles goes like that. They burst, they multiply within the red blood cells, and then they burst and they cause hemoglobin urea, the hemoglobin in the urine, and the fever, high fever is there, and hemoglobin urea is there. The hematological profile goes down, and they have some apical organelles. These are the apical, the front, the forehead of the parasite, and they have some organelles. That is why it is apicomplexan parasite. All the parasites have these organelles inside. So we are maintaining the in vitro culture of this parasite in our laboratory in a continuous mode. And this is the parasitic disease. The main thing is that once the animal is infected, it becomes carrier throughout its life. There is no drug till today which can clear the drug from the animals, and animal will remain the carrier, and it will go on spreading the disease to the other animal. So that is the main bottleneck. And the main important thing is that our export and export is affected. The positive horses with this disease, they cannot participate in any equestrian event. So that is the main part. And this is the how the life cycle goes. These are the ticks, you understand well. These are the parasite. So they have the two stages. This is the asexual stage, and all the sexual stages are in the ticks. And these ticks, through saliva, when they bite the animal, they introduce the sporozoites. These are the sporozoite, and then the sporozoite, they turn to the merozoites, and it is how the life cycle goes. And then again they suck back the merozoite stage, and they complete parasite complete its sexual stage in the red blood cells. And disease is very quite prevalent throughout the world. In India is very highly endemic country. You can see this, the yellow. This is the highly endemic country, and these countries are these are free. These are USA, Japan, Australia are free, and these are the coldest because ticks vector is not there. That is why disease is not in endemic form. And this is how these are the reason in India also. It is very quite widespread in India also. This we have been done the zero surveillance, and more than 50% of our equine population are carrying this parasite. And this is how the economic losses are very huge economic losses, because the drugs are very limited to this disease. And the carrier animal, they act as a nidus. They act as a nidus, and they go on spreading the to the other equine population through ticks, and this is how the parasite spread. And the infected animal, they show low performance, because equine is known for its performance and it low performance, and they become physically immunologically weak, and then the parasite proliferate or come on the scene when there is any depression. So right now there are only two drugs which are available in Indian scenario or in the world: imidocarb, dipropionate, and immunogen. These are developed in 60s, and they are very highly toxic drug. But these are the drug of choice. This imidocarb is the drug of choice, and till then there is no other drug which is effective and in use. So we are thinking of developing some new molecules, identifying some new molecules which can be taken further as a drug molecule. And there is no vaccine available, because this is the multi-drug resistance, and though no vaccine is animal, and it is very difficult to make the disease-free zone or make the animal to free of disease. That is why there is a need to develop some new drug and identify some new novel molecules which can be used. So we know that ethnoveterinary approach in drug discovery is very old, and we have a very high repository of the medicinal plant. And artemisia, it has been exploited more for the plasmodium, and we have artemeter, and many artemisinin has been identified from this plant, and this is the mainly Chinese herb. But in India also we have this Artemisia scoparia plant is available in India, and it is also a source of artemisinin. So we took this plant source, Artemisia scoparia, as a source, and this is the our lab strength, that we have many more than 20 or 22 plants and their extract we have tested in our lab, and we have identified these two plants as a potential which lead having potential: acacia nigrolica, this is Nina, and kikar babool, we know it in North India. I don't know what you know in the South India. And its bark, we have identified some molecules, and then we are working with the Artemisia scoparia, and this its leaves, we used its leaves in this study. So here I will restrict my talk to the Artemisia scoparia in this discovery. So we are just with the two objective, evaluation of the growth inhibitory efficacy of scope area, and then identification of the lead molecules in this molecules. So this is how we go the flow chart. We collect the plant material from the natural habitat, prepare its methanolic extract, and then we again fractionate the further with the we go from the non-polar to the polar. N-hexane is the non-polar and the methanol is the polar plant. So this is how we go from the distilled water is the polar plant. So we assume that the it depends on the solubility of the lead molecules in which the lead molecule is soluble. Some lead molecules are soluble in the non-polar and some are soluble in the polar. And then we take the extract from each of these and then check their anti-therial activity, because we have the mass in vitro culture in our laboratory, and then we check, and cytotoxicity we check on the PBMC, and then the most potential fraction was taken further with the TLC and with the HPLC. We again make it further fractions, and then we identify the lead molecules by the LCMS analysis. This is how the flow chart will go. So we prepared the extract. This is the methanolic extract, and this is the rotary evaporator, how we evaporated and make the extract the soluble. And then we can see in our case recovery of the plant extract, and the methanolic has 7.5% is the maximum recovery of the plant extract, because it is not like that if you take 100 grams of the plant all the molecules will be taken out. So 7 point or more 8 to 10% we recover plant molecules, so 7.5% we recover, and then make the serial dilution of these plant molecules and then test in the in vitro culture. And then in the initially we all the fractions were taken, we named them ABCD, and we see that the methanolic fractions is having the parasite inhibitory efficacy, and its IC50 is also very good in this case. If we see the PP, this is percent parastemia. We count the percent parastemia when we do the in vitro culture. We just put the drug at 1% of the culture and then see if it is able to inhibit the parasites in the culture, as I shown you in the first or second slide. And then we can see that the higher dilution of the cultures are showing the. So we take further this methanolic extract, and then we did the fractions with the N-hexane, chloroform, ethyl acetate, and methanol. And then we again fractionated it, and then we see that IC50 value is very less. IC50 value is like that the 50% of population is inhibited. So we can see at IC50 is 70 microgram per ml, whereas the so we can say that the fraction or the lead molecules or bioactive molecules are coming in the polar compound. It is polar soluble molecules which are coming in the polar 70%. And then we see the cytotoxicity. I don't know why the graphs are not coming. I'm not sure. Then we took the cytotoxicity of this plant on the horse PBMC cell line. We just take out the PBMC from the horse. I purified them, and then we seed the culture, and then see if it is toxic to the site or not. Then we can see the selectivity index is the very high for methanolic. When these fractions are toxic to the selectivity index is like that. If the selectivity index is higher, it means it is safe for the mammalian cell and for the mammalian cell, because the parasite is in the mammalian cell in the RBC. So we test it on the mammalian cells as such. If it is toxic to the mammalian cell, then there is no way of taking it further. So we see that the selectivity index is very high in our case. And then again, if the selectivity index is there, everything is fine. Then we want to see if they are toxic to the mammalian organ or not. Then for that, we just take the mice as a whole, and we have to calculate the LD50 dose. LD50 dose divided by 10 is the therapeutic dose. If you see this one, so there is a way of guideline are there, OECD guidelines are there to how to calculate the LD50, and then we come out that the the LD50 dose is here is around this one. and then we calculated LD50 dose at 2500 milligram per kg, and it is divided by 10, it is 250 milligram per kg. So this is the therapeutic dose. So 250 is the therapeutic dose. So we make the grouping. Three group we make. One is therapeutic dose, and one group is the half of the therapeutic dose, and one group is 2 times of the therapeutic dose, and one group is only the PBS. So four groups we took, and then the mice were taken. I think 9 mice were taken, 6 mice were taken in each group, and then we injected our extract, methanolic extract like this, and then we collected the blood at different time interval and see the hematological and the biochemical organ tests. We have defined liver function test and kidney function test and some other organ function test to do it. And then, of course, the mice were also sacrificed to see the histopathological changes also. And these are the, if you know, these are the liver function tests, SGOT, SGPT. This is the BU and the blood urine nitrogen. This is total protein, albumin, and the blood glucose. So we see how we see that this thing. So we can see that the group 2 mice, they are the different doses, and then we see that the in the group which is having the therapeutic dose, we can see it has very less effect on the organ. When the group 3 mice, there are 2 times of the therapeutic dose of the LD therapeutic dose, we can see that it has more effect on the organs. So this is how we can address the organ function test of this drug. So we see that the it is safe to the mice when we do the organ function test. So how this is how we ascertain that the methanolic extract that we selected is safe to the mammalian cell, and it has no organ toxic. So this is how we ascertain that this is the drug, or we can take it further for identifying the lead molecules in this methanolic extract. So this is how we now take it further. So identification of fatty molecules in the fraction D is the methanolic fraction, and then we perform the HPLC because we want to fraction it further, and we collected many fractions, and then with the 60 minutes the HPLC was run, and then we calculated, and then we collected the samples, and then the different peaks were collected. Around 10 peaks, 9 peaks we collected, and then we pool all these peaks, and then we freeze dried and to make it concentrate, and then we see how each peaks were again taken to the mass culture to see if these peaks are able to have any Theileria coli or anti-Theileria efficacy or not. So all these peaks were collected, they are pooled, and then they are freeze dried and concentrated, and then again taken to the in vitro inhibitory efficacy. This is how we take. So we can see that uh, these 3 peaks, the first 3 peaks, they are able to restrict the multiplication of the Theileria coli parasite in the in vitro culture. This is the percent parasitemia. The first peak is very good, second peak and third peak. Above that, we cannot observe any change, or they are the control is 7% and they are 6%. So we again pen down, squeeze the fractions to the only 3 peaks. We are now concentrating on the 3 peaks to take it further for the LCMS, or for identifying the lead molecules, which lead molecules are there to identify the lead molecules in this peak. So we submitted these 3 peaks to the LCMS analysis, and then because we are expecting the standards were already there, artemisinin, dihydroartemisinin, caffeic acid, the 3 peaks, we are just wondering if they are there or not. So we can see we collect we different spectra in the 3 peaks we observed. We can see we have observed the artemisinin peak, dihydroartemisinin, artemether is also there somewhere, and this is in the second fraction. In the second fraction we also observed the caffeic acid is also there in the second peak, and in the third the same the caffeic acid, artemether is also there in the third peak. So this is how these are the mark spectra and all the three peaks. Then we pen down. Then they are dihydroartemisinin, artemisinin, caffeic acid, artemether. These are the three compounds which are, and then we obtain this from the commercial, from the sigma aldrich. We obtain this commercially. So these are the four molecules that we procure from the sigma, and then we tested all these drug molecules again in the in vitro culture, and we see that IC50 is the main parameter for calculating the efficacy of the parasite. We can see that artemether is able to restrict the parasite at very low dose, and and this we also do the viability test. If we inject the drug, then we want to see if the parasite are viable or not, because if the parasite may also restrict its growth. So we see that the parasite are not viable with the artemether and with the artemisinin, and artemisinin is effective up to 20 microgram drug concentration, and it's also very good. And dihydroartemisinin is also very good. The caffeic acid IC50 is little bit more, and the parasite also viable after the drug treatment. So we can say almost the three drugs are very effective. The artemisinin, artemether, and dihydroartemisinin are very effective out of the four molecules. Because of course the spectra was very big, but we were only able to identify the four molecules, but other molecules were also there, and that were for that the next library was not with us, so we are just processing the data to identify some more molecules so that we can test these drug molecules in our system. Then we worked on the drug target. Which are the drug target? Because these are the drug already known to inhibit some of the plasmodium, and from the blast search we identify that dihydrorated dehydrogenase is the drug target for this, because we have to establish how the mechanism of this drug, how these drugs are restricting the growth of this parasite in the Thaleria equi. So we took out this drug, this target from the Thaleria equi source, and then we prepared the homologous model of the dihydrohydrogenase reductase as a drug target for the Thaleria equi, and then we demonstrate that it is inhibiting and it is restricting the growth of the amino acid, is interacting with the amino acids of this parasite, and this is how we can see the common amino acids are there, because we make one model and then we did docking analysis with the artemether, artemisinin and dihydroartemisinin. We can see the alanine 74 is also it is also there. Phenylalanine 366 is also the amino acid. It is being blocked by the artemether and artemisinin collectively. So there are common drug targets are there which are acting against this parasite. So it conceptualize our hypothesis. Yeah, these drugs are there and they are blocking this. This is how we conceptualize the mechanism of action of this drug. And dihydroartemisinin, caffeic acid. You see, the caffeic acid is very weakly bound to this drug. That is why it is not effective in inhibiting the growth of this parasite. And this is same phenylalanine 366 is also able to block, is again there. So they have the common amino acids which are restricting, which the drug is acting on these amino acids, and they are it's a common mechanism of action, these three drugs, and the caffeic acid is not so effective in restricting the growth. So this is how we come to the conclusion, and this is the database identifying their medicinal value from the database itself, and these are the references. Because references are available, and these are the other medicinal value of this drug. Quercetin we have also already tested, but it is not effective in our in the Thaleria, because camphorol is also not effective in our system, because these are quercetin, camphorol are oxidative stress reliever, but they are not effective in the Thaleria equi as such. these are some other database from the database. So in conclusion, we can say that all the fraction of Artemis square except fraction B were effective in inhibiting the Clear-acquisition growth. And the fraction D, which is the methanolic extract, it's very effective, and its IC50 value is 70. And the HPLC, we observed the and LCMS, we have penned down the 4 drug molecules: artemether, dihydroartemisinin, artemisinin, and caffeic acid. And the DOCIC analysis confirmed that dihydrogenase reductase is the drug target for this drug molecule. This is how we penned down we completed our hypothesis for this. And of course, we are collaborating with the some this is our campus at Hisar. So I just finished my talk. If any questions are there, we can discuss. And this is the NRC on equine, just about the NRC on equine. This is our mandate to do researches on the health and production management of the equine and to develop diagnostic biologicals for major equine disease. So our center is working on the various viral, bacterial, and protozoan diseases and to develop the vaccine and some biological drug against this one. And we have the programs and activity and technologies were generated by our center, many centers, and we have the farmers activity also. And they are the list of the vaccine or the diagnostic that we have developed: the herpes virus and the trypanosoma, if you heard, the glanders is also rotaviruses, and the vaccine we have also developed against the herpes virus infection in dog in equine, and the rotavirus we have developed many vaccines also. And these are the some acknowledgement who are associated with me in this ARV. We did the HPLC work. The student belongs to the Indian Veterinary Research Institute. He was my PhD student, and ICR is my parent organization. So this all. Thank you very much. I think I was not too fast. We can discuss more.
So thank you so much, sir. This was a very knowledgeable session on the phytochemicals and molecules found in Artemisia scoparia and their antithylaria effects. So thank you so much for taking
Thank you very
your
much.
time.
Thank you. Thank you.
So if any of you have any questions, please feel
Any
free to
questions
post
there?
them.
Yeah.
So, sir, it looks like we don't really have
Any
any
questions?
questions. Mm.
Okay. Thank you.
So, yeah, thank you so much, sir, once
Thank you.
again.
Thank
It
you.
was a
Thank you.
pleasure having you with
Thank you.
us.
Thank you to being with part. Thank you very much.
Thank you, sir.
Thank you.